and thank you for the invitation to this conference. And I'm really happy and impressed by the organization, and I'm very proud also to be here today talking in this the very first ever EvolTree Conference. And I wish really a very long life to this initiative. So I will be talking today about how we can use genomics to improve predictions of population responses of forest trees in the face of climate change. And what I will present today is mostly the work of a PhD student that is with us here in Bordeaux, co-supervised by myself and another researcher that you may know because she has done big contributions to this topic, Marta Benito Garzón. So the student is named Juliette Archambault, and I hope you will like our talk. So I will start just presenting some our actual context in Europe. So probably all of you have heard some talk about the European Green Deal. This is a political initiative for the European Union to move to a green-based economy. For what we expect this talk, one initiative that is very important is the European Union biodiversity strategy that is planning to plant an additional 3 billion trees by 2030. That means that what is requested for us for research is to provide tools and ways and insights and background research that could help to do this big effort for this big effort into plantation and restoration to be successful, in particular by matching the best provenances with the best sites for each one. So how we can do that? I think one very interesting approach is that it has been already used in the past, but we will be in more and more use, I believe, is to use models to assess the population responses to future environments of different genetic materials. One thing that we are lucky in forestry is that we have a long history of common gardens. So they are this is a discipline where we have done a lot of work and a lot of effort and investment in this comparative plantation, which have provided the background information for and the data to develop these modeling efforts. And I think most of you will be familiar with this kind of figure, where where we have models have been produced to try to estimate what could be the performance of different genetic materials when they are planted outside the local population. So, and there is a number of these models, as you know, and what we will be dealing with today in my talk is mostly about how we can integrate genomic and genetic information in these models. I mean, information coming from molecular markers to try to improve the predictions, mostly in cases where we don't have the target provenances planted in common gardens. And we believe that genomics can help in these cases, and I will show you some case study and some literature on that sense. So what would be the advantages of including genomic data in predictive modeling? First, we will help us to understand better what are the drivers of these genetic responses of forest trees to new environments. Then they will help also to integrate the interspecific variability of that is quite big in forest trees because they are undomesticated species, and a scale that is finer than the population. We could look to genetic variation at the individual level. as I said, it can help to produce better predictions and to make these predictions more accurate when we are trying to deal with provenances and species that for which there are not a lot of information, a lot of common garden. That is actually most of them. And then by using genomics, as now it's relatively easy to produce genomic data, we also increase the chances to produce a rapid response to the request that they make for us for applied operative forestry. So how we can integrate genomics? So I will start from the most simple way is just to consider distinct different gene pools. And for instance, this is a very interesting case study where could see if I can. Yeah, I can use a pointer. So this is just based on niche modeling, what is classical, what is the first kind of models that came out to deal with this kind of issues. And here what you have is just some models of the distribution of a bat. I mean, it's not a forestry, it's just it's a bat, it's a forest bat, but still a bat, an animal. And then you can see here what are the predictions when you do models of the future distribution, the present and the future distribution of this bat, using just all the data together, all the individuals together, all the populations together. And you can see that there is quite a strong predictions on the evolution or the distribution of these species. But then in this study, what is original is that they did an application where they used molecular markers to identify different ecotypes in this species. And they found two different ecotypes. One that was was inhabiting cold environments and another hot environments. And then what it came out is that when you do the modeling for the different ecotypes separate, then you will have predictions that are radically different from when you do the predictions combining the data. And it's what you can see here in this cartoon, where actually the reduction of the distribution is going to be actually too big when you consider these genetic differences at the interspecific scale. And this is a very simple way of using markers or genomics into models. And I believe we believe it's really important in forestry, as we have really several studies that are showing that most species have substantial population genetic structure, and we could work with differences. We could introduce these differences quite easily in the modeling approaches, and it has been done already for some forestries. Another different way of using genome information that is a bit more complex, and it could be by using established genetic and environment correlations to do predictions for vulnerability or maladaptation, what is called the estimation of the genomic offsets. I'm not going to talk too much about this because there have been already one talk and some poster about it, and there will be another one coming in this session after my talk. And it's a approach that is becoming, I think, very popular, and it's been applied to forestries and all kind of plants and animals. I brought here an example in a migratory bear that is using this approach, and it was all based in a landmark paper by Matthew Fitzpatrick and Stephen Keller that brought some communications to this conference, so you will hear from first hand how this works. And this was in 2014, and in this just the last 7-8 years, there have been already several applications. And my message about this method is just like, it's not so simple as it looks like. What is more important is to do a good validation. And for that, we have two different ways. One way could be to use inventory data to validate what the predictions from this genomic offset or for this distance in allelic composition of the current population from that could be optimal in the future. That is what the genomic offset is trying to evaluate, but it needs still validation to bring it to the operative level. And apart from field inventories, another way of validating that you will, there will be a talk about this, as I say later on, is using common gardens for seeing if this genomic offset is truly related with the difference in phenotypes and fitness. So, and I take this like for this use of the common gardens and validations actually to highlight that as foresters and people working in forest genetics, actually we have one of the few advantages we have that we don't have many of them, but one that we have is that the existence of common gardens. And common gardens can be used not only for validating things like genomic offset, but also it gives us access to traits, direct access to traits, and that can provide some very interesting data that we can use not only when we genotype these common gardens, we can use it not only to build models that predict directed traits based on the phenotypic measurements of common gardens, but we can use also the genomics to do trait prediction. So this has been done in two ways, like for doing the predicting phenotypes of based on common gardens. One of them is what is called the polygenic scores. That's just a way of integrating all the markers that are associated with the phenotype in a single score. And what is relevant of this polygenic score is that you combine the information of, you take all the markers that are associated with the trait and you weight them by the estimated effect on the phenotype or the estimated effect on the trait, these betas. And this can be very complicated. So, but it's a method that is becoming to be used also in forestries, and it's attractive because if allows for accounting for different size effects of different polymorphisms. And in a way that is simple and it seems also to be quite effective. They have already there are already some applications that are quite interesting. And I will explain during my talk the case study we have developed in Maritime Pine for this is to use positive effect alleles. And the positive effect alleles is just simple counts of the SNPs that have no zero effects on phenotype. You just count the alleles, the positive alleles, the alleles that are associated with a particular phenotype, and in principle will give an advantage to the populations or the trees that have this. And this is the two ways of integrating or replacing phenotypes by genomic information. Both of them rely on GWAS. That means that we need to do a previous step that is a genotype-phenotype association. And given that what we know now from model species, but also from forestries, that GWAS normally identify multiple loci with very small effects, but that is very hard to find really causative markers that have a strong impact on the phenotypes. This claims to use approaches that they are based on polygenic association, rather that try to identify the candidate gene or the loci that could have big impacts on traits. Here in the cartoon, you can see a review by Brandon, that he was one of the speakers in the first day of the conference, and you can see for different kind of traits what we the kind of size effect we have in association studies. And I said they are lower than, they spend lower than 5% of the variance. So really one single mutation is not telling much to us. But what we are learning in the few case studies where we have applied polygenic association for the trees is that actually you can explain quite a substantial amount of variability and or the datability of the trait when you use you combine multiple markers in the single model. And where we had learned in that sense in the studies we have done in mite and pine is actually that when we look to polygenicity for a wide variety of trees, and you can see here some different category of traits, and these are estimates of polygenicity defined like the percentage of markers that have an effect on the phenotype. And we are in levels of 5–10%. That means that about what our estimate says is that about 5–10% of the genome is associated with a trait. And what we have seen is that these effects, they are all very small and they are all very similar. And actually, when we look to the combined effect of these SNPs, it's basically the datability we managed to explain depends mostly on the number of loci rather than the average effect size. And this is, at least in the case of mite and pine, that is the one we are developing. This is claiming that we could go, actually, when we use positive effect alleles, we are collecting actually a very similar information that we got for more complicated polygenic scores, because the size effect of the loci are more or less the same. So we don't need to, actually we simplified a lot. If we just count these alleles that are giving positive, that have a positive impact on the phenotype, and we skip the step of estimating the size effect, the effects, the betas in the polygenic scores, because they are always very complicated and subject to error, because we are estimating normally many parameters with a small data set. So okay, so with this I will present our case study. This case study is based in 5 common gardens, and 3 common gardens under Atlantic climate. Is like for mite and pine, that's very good conditions for growth. And then we have another 2 common gardens that are under very harsh Mediterranean conditions. That means that are in an environment where survival is difficult. And then we have this is quite a large data set for the phenotypic information. We have about more than 30,000 observations of height in 34 provenances, and this is a clonal common garden. So this is all repetitions of 523 clones. Working with clones has also the advantage that it allows for very precise phenotypic estimates. So I would say that we have a very good phenotypic data set, and then we have quite a small, for what it is today, a small SNP data set for just 5,000 SNPs. So, but actually, we believe our own resource that we have high levels of polygenicity. That means that for these 5,000 SNPs, about 10%, that's it, like 500, will be involved in the control of height, or at least correlated to link to SNP that are associated with height. And one other relevant thing you need to know about mite and pine to be able to understand our case study, this is a species where there is a lot of provenance variation, and it's a species where there are a strong population genetic structure with gene pools having differences in markers. There are neutral gene pools, they are different, but also they have strong differences in growth. And this is some models just from this common garden showing our estimates of growth for each line here is a population, a provenance. And each distribution, the color represents different gene pools, and you can see that there are gene pools like those in here in some Western France, in the Atlantic coast, that grow very well, most of the populations, and there are others like those in southern Spain or Morocco, they are not doing so well. So, but there's a bit of variation also within the gene pools, but this is an important factor. And you can see here, this is the one common garden we have here in Bordeaux. These two trees, they have exactly the same age. So you can see that one is more or less half the size than the other. And then this one, the small one comes from Morocco, the big one comes from here, from the Landes region in southwestern France. So what we did, we took this data from these common gardens, and we built 12 models that will, in these 12 models, they were based in the standard modeling like we do for doing standard quantitative genetic analysis and transfer rules and the standard analysis, but then we were adding a few additional parameters. So basically what you have to keep in mind is that there are some baseline models here, that this is just the standard models that include the site, the trial site, the provenance, the clone, and the interaction. That's just quantitative genetics as usual. And then there is a number of models that they are introducing the climate of the sites and the climate of the provenances, that are the traditional or the standard modeling. So that we have been doing already to this work by Wenfer and others, where they are looking to these transfer rules, the cartoon they showed you in the first slide. And then what we are trying to do here is to add to these models, that they are other models where we take into account either a combination of climate and genomics, or just genomics, and see we can replace climate, the information about climate, or the climate of the trial sites and the climate of the provenance, or the original location of the provenances by genomics. Or if we could combine different factors, we get a better model actually that in predicting the trait that we will get just knowing the standard quantitative genetic analysis. And then we did these 12 models, and then after doing that, well, we will compare them using data partition and cross-validation, and compare the predictive errors as well. And the cross-validation was very important, because what we wanted to know is, like, using the genomics, we can predict provenances that are not included in the common garden, that will not be really in the common garden. So that is actually I think the biggest application of this kind of work for operative forestry, if we can do that by using genomics. And what is new is as I said, for one side, is that the introduction of the genomic component, and based on these positive effect alleles, that this is affecting a number of models. And also, as maritime pine is maritime pine, and it has this strong population genetic structure, we also included the information about the belonging to one of the gene pool, like part of the model, to see if just by knowing the gene pool, we get already some information on the phenotypes or better prediction. So we did these 12 models, we compared them, and then as this talk is about mostly how to introduce genomics into the modeling. And so basically to do that, what we did 4 different kind of association. First, we did a standard GWAS, a standard association between using the 5 common gardens. And when we use these 5 common gardens, that means that we are including in the association model 5 different environments. So when we get a significant association, we are finding SNPs actually that have an association with height, with the trait we are targeting, in or the distribution of the species, because these common gardens are covering the environmental variation of these species. And then we thought, well, this gives us something that we call global or range-wide allelic effects. But then we did another thing. We took common gardens by environment, and we put on one side the Iberian Atlantic, then the French Atlantic Common Garden, and then the Mediterranean Common Garden, and we did independent association studies for each one of these common gardens or group of common gardens. And this is giving us the SNPs that are associated to the trait in different environments. And they have a regional value, to say. They are environment specific. And then we build models either considering these global positive FR alleles or these regional or environment specific positive FR alleles. So we got our best model, that is this, and is model number 8 or the 12, actually as predicted, as we thought would be, is including information about the gene pools. So this is the intercept for the gene pools in the model. You can see that there's a nice gradient. Different gene pools has clearly different growth, and then gives really, just by knowing the gene pool, is giving already some information for prediction. But then probably what is more interesting is that also we found a good, in the model, we also found a quite good predictive power by the positive FR alleles in the different common gardens. When we take the 5 common gardens together, when we take the specific, the regional, the specific, the environment specific positive FR alleles, and this has has an impact in the precision of the models, and also as usual we got some climatic variables. So the best model actually includes climatic and genome information, and this genome information is both the gene pools, the population genetic structure, to which gene pool the trees belong, the populations belongs, and this regional or environment specific positive alleles. But what is interesting is that it doesn't include the common garden design, the provenance and the clone effect. So we managed to replace the information from the origin of the materials and the plantation sites just by the genome information, the climate, and the climate information. And this is quite encouraging for future applications. So what is more, then when we look for predictions, how good are predictions? Because I say, well, okay, you have, this is the best model, but how good are the other models? And you can see that for the just to remind you, we have this baseline model that is just quantitative genetic analysis as usual. Then that is this red and orange colors here. Then we have the models that combine climate information and genomics, that is the green ones. And then they have the ones with only genomics, that is 2 dark blue here. So we can see that there are a number of common gardens. This is we did it separate for the 5 common gardens. So we have those common gardens that are in a good environment, like the one in northern Spain, in Bordeaux, and in Portugal. Actually, there is not a big difference between models. That's already positive, because we see that a model with only genomics is as good like a model with climate and genomics, and as good as the quantitative genetic model. So we were happy. But what is even more interesting is that if you look to the common gardens that are under harsh Mediterranean climate, what we see is that we can predict better actually with the models that include climate and genomics in the 2 of them. It's a bit better when you take the 2 together, but actually when you take only genomics, is working also quite well. So it's not so bad. So and this is really I think the most interesting part of my talk, or like of how, like, by using genomics, we have the potential. Still, we are still validating this result, but if we have a good GWAS study based in a proper association panel that represents well the population structure of the population, we are able to do a prediction of quantitative traits in provenances that are not included in this common garden. So, and that's my message today to you. So, and then it was also interesting for us to see how it's actually then when we compare the model based on genomics in positive alleles, we were not sure what we will come out, but we saw that actually the model that are including the positive alleles identified in particular environments is better. It's quite better in prediction that the model that is based in just this in SNPs that will have a global impact on all kind of environments on the phenotype, and that comes quite well. That goes well with predictions about conditionally neutral alleles. This is a discussion that probably you are familiar with between two modes of adaptation translated to the molecular level. That is the antagonistic pleiotropy, saying that if you have a gene or a loci that have important for adaptation, you will have one allele that gives a positive fitness, the opposite allele will give negative fitness. So you will have really the two antagonistic effects depending on the allele. And but there is another theory that is the condition neutral theory. They say, well, no, there are number of genes of SNPs that will be neutral under a particular environment, but then they will have an adaptive value in another environment. So you can see here how this under condition neutrality, this marker will remain neutral in this range, but then they will produce a selection differential in this other range. While under antagonistic pleiotropy, you will have an allele that will produce a negative selection differential in this environment, but a positive in this other one. And actually, our result they give a little bit support to this condition neutrality model and also could be because we could get this result just because we also the conditional neutral alleles are more difficult to identify in global association. So it could be also some trick done by the way we did the GWAS. But in principle, we have explanation for these differences. And with this, I will move straight to the conclusions because I don't want to. I know it's Friday, and this has been a long conference, so I don't want to take a lot of time. So we have a lot of time for discussion and questions. So I would say that my conclusion is that the best predictions we got is when we combine the population demographic history with the provenance climate of origin, and when we incorporate also this positive effect allele associated with height, and that's our best model. And then we found again these positive effect alleles seems to be very often specific to a particular environment, and this is not very good news actually, because in a way that means that we will need to have common gardens in many environments to be able to identify the relevant variation for a particular trait, being able to use it in modeling. And well, this resource is supporting this conditional neutrality model. And what for me was very, very interesting is that that actually even models only using genomic information were as good as models based on the common garden design, or even as good as models using climate information. So this is still to be validated in other species, because that can maybe something very specific of mountain pine, and that is a species with quite a strong population genetic structure. But the population genetic structure is not fully confronted with the phenotypic variation and with the environmental variation. So it's a very actually promising and encouraging result. And then as some, just to finish, I would like to give some general perspectives. First, that we need, for everything we do with genomics and models, that we have any application to tell forester where they should plant what, we need a lot of validation. I mean, there are many statistical uses with these methods and with the modeling that is associated, and we had to consider that very often we are extrapolating to unknown climates and to unknown populations, and we can go further and say unknown biotic interactions and many unknowns. So everything we can do in terms of validation could be very good, and we need to do this validation pretty fast. We want these approaches to be useful. Mother message is that we started the work thinking, oh, maybe we could go and produce something that would be useful and will allow us to be independent of common gardens. But actually, what we are seeing is the contrary. We really need the common gardens. So either to train the model, we need common gardens. To validate the models, we need common gardens. So also we should do a strong effort in planting more common gardens for the future. We know that common gardens are difficult to plant. They are expensive, but we need them. We need them if we want to be serious about predictions and to really produce information that is valuable. And all I've been talking about is about polymorphism, SNPs, but there are other sources of genomic variation that are also related to phenotypic variation, adaptation, like copy number variance, etc. So, and this also information on expression that has also a lot of interest for prediction and could improve the accuracy of the models of the kind we have been showing here. And with this, I just wanted to thank you for your attention, and yes, I will take any questions you may have. And also, if you are interested in this kind of approaches, both Juliet and I will be teaching in a shared EvolTree before this course that will take place next spring, as Felix was kindly presenting during the session at 1 p.m. And so you are all welcome to join and join us in working with these topics.